So what I'm going to talk about today are some aspects of pathogenesis of hepatitis C with special reference to liver cancer. But I wanted to start off by placing hepatitis C in the context of other hepatitis viruses. And Mike is absolutely correct. When I graduated from medical school, there were two hepatitis viruses, hepatitis A and B. And it was only with the development of good tests serologically to detect hepatitis A infection did we realize that there are other forms, in particular non-A, non-B hepatitis, now primarily known as hepatitis C, the old post-transfusion non-A, non-B. But I show this slide to make the point that there are these 5 viruses, all of which have tropism for the liver, and all of which can cause acute hepatitis, even hepatitis C occasionally. But these two in particular, hepatitis B and hepatitis C, have this tendency to cause long-term persistent infections that can lead to cirrhosis or scarring and fibrosis of the liver and liver cancer. And this is one of my favorite slides. I'm not sure that the data are accurate, but I like it because of the way it shows things here. This is from an article in Nature Medicine about 6 or 7 years ago that compares the global death rate due to viral infections and other causes. And if you look where hepatitis B and hepatitis C show here at a level of 6 on a Richter scale, and I imagine that's a logarithmic scale, we're talking about an equal number of deaths as road accidents, somewhere between non-HIV TB and malaria. So these are important infections. I think you've already had a good lecture on virology of hepatitis C from Steve Polyak, so I'm not going to spend much time talking about the virology, but I do want to remind you, and maybe some of you weren't here for that lecture, that the virus responsible for this disease is a positive strand RNA virus that's classified in the family Flaviviridae. It's an envelope virus. And it's a virus that doesn't yet have any clear-cut morphology that we have yet been able to really identify, but this is probably a picture of a virion with antibody to E2 decorating it. It has these 2 envelope glycoproteins that are heavily glycosylated, a nucleocapsid protein that is part of the virus structural protein complement, and then it has these 5 non-structural proteins that are involved in replication of the viral RNA. And these 2 other proteins, membrane-associated P7 and NS2, that play roles in assembly of the virus particle. Now, one important point to make at the outset is that when you have a virus such as this that has a relatively small genome, less than 10,000 bases in its entirety, and expresses only 10 mature proteins, and yet has to do all these things, figure out how to enter a host, replicate, make new virus particles, and get out, and all the time evade and escape the immune system, it's only natural to understand that each of these proteins probably carries out multiple functions. There's more than 10 functions that are required to do all that. So a hallmark of these kinds of viruses typically is that any one of these proteins could have 2, 3, half a dozen different functions. And part of the fun of working in this area is to elucidate those different functions and to try to relate them to the sequence and structure and pathogenic potential of the agent. So the life cycle of the virus, as we understand it, is completely cytoplasmic. The virus enters through attachment to specific receptors, a process that's being elucidated in a number of labs and is proving to be really very interesting as it involves a number of tight junction proteins, and releases its RNA into the cytoplasm, which is then translated, leading to the expression of these structural and non-structural proteins, with the non-structural proteins assembling on intracellular membranes, probably derived from the endoplasmic reticulum, and forming these replicase assemblies that are located within very small vesicular structures that are known euphemistically as a membranous web. And that's the site of RNA synthesis and probably also assembly of the particle, which is then released in 2 manners, one directly into the extracellular fluid, and probably also by a mechanism we don't totally understand that allows the virus to pass to another cell without ever seeing the extracellular milieu. So when we talk about cancer, as we will here, I want you to remember that this is a virus that never sees the inside of the nucleus. And obviously, cancer is a nuclear process. So something to think about. So the natural history you've probably heard about also from Merriam-Alter, but it's perhaps worth going over again. The hallmark of this virus is its ability to achieve persistent infection. And that happens probably in 60 to 70% of individuals who are infected with this virus, even if they are fully immunocompetent adults. So that's very different than hepatitis B. You might read in textbooks that hepatitis B causes persistent infection 5 to 10% of the time. Well, that's not so in immunocompetent adults. Persistence with hepatitis B is probably less than 0.1% in that population. But in hepatitis C, it's quite different. This virus has developed ways that I think you'll hear more about from Mike and maybe some other speakers later in the course that allow it to escape both the early innate and late adaptive immune responses. There is a period of peak replication, and then it sort of drops the amount of virus in the blood to a rather stable set point, and it persists there for years. And during that period of time, one notices in the majority of infected individuals, those that fail to clear the infection, fluctuating alanine aminotransferase levels. This is an enzyme that's released by injured hepatocytes and is found in the blood and can be tracked in the blood as a measure of hepatic distress or hepatic liver inflammation. And then if you look at the liver, you see a progression from a early period where there can be quite a bit of necrosis and inflammation and associated fat, steatosis, and we'll come back to that later, to a picture that's typified by progressive fibrosis, with scar tissue being laid down within the tissue of the liver, extending across portal triads in a fashion that's known as bridging fibrosis and eventually leading to cirrhosis. That is what kills most people that die as a result of this infection in the United States. It can take perhaps 30 years to get there, but in the US today there's probably on order of about 10,000 deaths a year due to cirrhotic liver disease associated with hepatitis C infection, and for which the progression of the disease may have been potentiated with alcohol. So it's an important disease, without doubt. Now, once cirrhosis has developed, as many as 2 to 3% of individuals each year will develop liver cancer. And that's what I'm going to spend most of the time talking about today, trying to understand this progression to hepatocellular cancer and why it has this special association with hepatitis C. Now, in the United States, there's probably 10 deaths due to cirrhosis for every 1 death due to liver cancer. But in Japan today, for reasons we don't totally understand, that ratio would be reversed, and one would see 10 deaths of hepatitis C-associated carcinoma versus maybe 1 due to hepatitis C-associated cirrhosis. So we can discuss pathogenesis of hepatitis A in several different ways. Keep in mind that the only cell for which we have really good evidence as being susceptible to infection with hepatitis C is the hepatocyte. As shown here, hepatocytes are present and arrayed in these plates of tissue that extend between the portal vein and the central vein within the liver, with the sinusoids on either side. So we can talk about how the virus infection perturbs the intracellular milieu and how it actually alters the intracellular environment. And we'll spend a little time talking about that today. Now, that can be accomplished by the virus in order to evade from an innate antiviral response that the cell is trying to evoke upon recognition of the virus. It can also be a change in the intracellular environment that the virus has caused in order to promote its own replication. And those are the changes we'll be talking about primarily today. You could also then talk about what I would say are system-level interactions of the virus. Innate immune responses at a system level rather than an intracellular level. Adaptive immune responses, inflammation, steatosis, activation of stellate cells that actually lay down this fibrosis within the liver, and malignant transformation. And then finally, we can talk about the host consequences, which would include hepatic failure, cancer, or cryoglobulinemia in a small proportion of individuals. So if you look at Japan in the 1980s, there was a dramatic change in the pattern of hepatocellular carcinoma, which has always been a problem in the Japanese population. Prior to 1980, and this is retrospective studies obviously, most of the cancer identified in Japan was due to or associated with hepatitis B, and a relatively small proportion hepatitis C. But beginning around the late 70s, as noted by Professor Kiyosawa first, there was a pretty dramatic increase in the incidence of death due to liver cancer. And that was not a reporting artifact, but a true increase in deaths due to liver cancer. And it was primarily among men, and it was associated with a shift from these cases primarily being associated with B to a pattern which by 1990 was by and large exclusively associated with hepatitis C. So a real shift in the dynamics of liver cancer in Japan, and that reflects the introduction and dissemination of hepatitis C within the Japanese population back in the late 50s, mid-50s, and into the 60s. For reasons that we can talk about perhaps offline afterwards if anyone's interested, the spread of hepatitis C in Japan antedated the spread in the United States by about 20 years or more. Now, in the United States, we're beginning to see similar increases in liver cancer. And these are data from the NCI from 1974 to 2004. And what's really important here to note is that the group that has the most rapidly increasing rate of liver cancer is the Hispanic Americans, primarily of Mexican origin, and African Americans. And these are the Caucasian Americans down here. And this relative difference that you see here in the incidence of HCC ties very closely to differences in the prevalence of hepatitis C infection in those populations, although we don't really understand why the Hispanic population, and again, it's primarily Hispanic males, is so much higher than African American males. So this is a problem I think we can all anticipate will be one that will continue to evolve in the United States unless we come up with better therapy for hepatitis C. And presumably sometime in this course, you'll hear more about what's available in terms of new approaches to treatment. So how exactly does hepatitis C cause liver cancer? Well, I think you have to consider, as you think about this, that any hypothesis must include the fact that there's a lot of inflammation within the liver in chronic hepatitis C, and this goes on for years, 25, 35 years prior to the development of cancer. A great deal of oxidative stress with reactive oxygen species being generated, and great potential for DNA damage due to that oxidative stress. And yet the emergence of cancer in the background of hepatitis C-related chronic hepatitis is greater than we see in other forms of autoimmune hepatitis, for example. So we can't say that it is simply chronic inflammation. We also have to explain why the risk of cancer is increased with even moderate regular ingestion of alcohol. And that might get back to a contributing role of reactive oxygen species and oxidative stress. However, as I'll show you, we know that transgenic mice that express various hepatitis C proteins also develop liver cancer. And they do so without drinking alcohol and in the absence of inflammation. So that suggests that hepatitis C proteins are in fact directly oncogenic. There is a clear-cut association with cirrhosis, and almost all forms of cirrhosis put individuals at risk of cancer for reasons that at least I don't understand. And that's something that needs to be considered. And we have to also consider the fact that HCCs, or hepatocellular carcinomas, as they develop, are genetically heterogeneous. Typically, there are multiple chromosomal reduplications, and there doesn't seem to be much of a pattern to that, although, as I'll show you in a few minutes, some recent data from Snorri Thurgerson at the NIH is beginning to suggest that there is, in fact, a very regular pattern to that may be of great importance. It's also worth just remembering what the other causes of liver cancer might be, for those of you that don't work in the clinics or think about this much. Aflatoxin B1, certainly, hepatitis B virus, Helicobacter infections, hereditary hemochromatosis, up to 30% of adults with that disease, and alpha-1 antitrypsin deficiency. many of those causing potentially cirrhosis and perhaps acting in part through this cirrhotic procarcinogenesis mechanism that we don't understand well. Well, I alluded a minute ago to some recent data from NIH that has looked at genetic changes with liver cancer. And this slide tries to summarize some of that data that's just been presented and published this year in recent articles in Cancer Research and in Gastroenterology. This work comes from Snorri Thurgesson at the National Cancer Institute. It's been known for quite a while that the progression to hepatocellular cancer is a multi-step process. You don't go from normal liver to liver cancer overnight, but what you do go through is a process from normal liver with fibrosis to cirrhosis to regenerative cirrhotic nodules, where the cells are obviously proliferating within a nodular fashion constrained by surrounding fibrous tissue, that can go on to develop low-grade and then subsequently high-grade dysplastic changes, and then finally progress into early hepatocellular carcinoma. The morphologic changes associated with this are really difficult to read for a non-pathologist, and it's I think, a fairly fine distinction, but one that pathologists can agree on. So what people have shown, and Thorgerson in particular, is that during this progression, there are accumulating genetic and epigenetic modifications to the cell that actually lead and account for the malignant transformation. And in particular, a genome-wide screen, as well as intensive use of oligonucleotide microarrays applied to considerable number of cancers of diverse etiologies, suggests that in this transition from the high-grade dysplastic nodule to the early hepatocellular carcinoma, the cellular proto-oncogene c-MYC plays a really fundamentally important role. And that pathway analysis suggests that in over 85% of those tumors, during this progression, there is activation of a c-MYC-controlled pathway, as reflected in this diagram here at the top. So that actually correlates with increasing c-MYC copy number with duplications of the 8q chromosomal region in these cells, and suggests that c-MYC plays a central role in the development of liver cancer of multiple etiologies, not just hepatitis C. And that's Snorri Thurgesson's data, and I'm not going to talk too much more about that. However, if you express c-MYC in a transgenic mouse model, these mice develop liver cancer. And if you look also in those mice, you'll see that in the hepatocytes, look at the third bar here, and this is again work from Snorri's lab that was published in Gastroenterology last year. There is both increased proliferation of hepatocytes, as indicated by PCNA labeling in the tissue, and also increased apoptosis of hepatocytes. So c-MYC overexpression in the liver promotes hepatocellular proliferation, also promotes hepatocellular death. Now, other work from Snorri's lab indicates that overexpression of E2F, which is another cellular proto-oncogene, it's a transcription factor that's regulated by the retinoblastoma gene, and we'll come back to that in just a minute, also causes liver cancer in mice. And it also causes increased proliferation and increased apoptosis as well. But what's important is that there is an interaction between E2F and c-MYC, in that E2F expression in double knockouts, double transgenics, excuse me, reduces the apoptosis that you see caused by c-MYC overexpression. So if you look then at what appears to be cooperative actions of c-MYC and E2F, both cellular proto-oncogenes, both of these oncogenes drive the cell to proliferate, and that's shown in the pathways over here on the right, largely through mTOR and increased cellular translation. But the combination of c-MYC and E2F also has an anti-apoptotic effect. And we understand more and more that the development of cancer requires both a drive for the cell to proliferate as well as a disruption of the cellular controls that cause the cell to die of apoptosis, often through p53, but other pathways as well. So bear this in mind, this interaction between E2F and c-MYC as we go on in the talk, and also what I mentioned earlier about the really central role of c-Myc in particular in that transition from late dysplastic or high-grade dysplastic nodules to early hepatocellular cancer. Well, I mentioned earlier that the virus hepatitis C, to come back to it, the perpetrator here, replicates within the hepatocyte. And I just want to show this one slide that comes from some recent work in the lab. Shown here is an elastin stain of liver tissue from an individual who actually had a liver cancer resected at UTMB. And what you see is these bridging fibrous septae that bridge the portal triads in the tissue here, what's left of them. And these are containing these regenerative nodules. This is with the conventional fluorescence microscopy. To the right here is an image with two-photon microscopy where we are staining the same tissue now at a higher magnification, the boxed area here in yellow, with an antibody that's specific for core. And you can see here in the regenerating nodule the presence of core being expressed by the virus. So we know the virus is right in those cells in the tissue in the regenerative nodules where there's dysplasia and where the cancer will arise from. And the question is whether the presence of those viral proteins have a direct oncogenic effect. And the answer for that comes from the study of transgenic mice. And I'm going to show you a few slides from a study we did quite a few years ago. This was published in 2002, back before we had any ability to replicate hepatitis C in the laboratory. So, you know, when you want to work with hepatitis C and you can't grow it, what do? You overexpress the proteins and see what happens. We overexpressed them in black 6 mice. What we did was to overexpress the entire polyprotein of the virus under control of the albumin enhancer promoter sequences so that it was liver specific in its expression. And we achieved several lineages of mice, one in particular, FLN35, that we studied in detail, that had very low level expression of the polyprotein, very difficult to detect, and comparable to what one would find if one looked in human liver. So what do we see in terms of pathology in these mice? Well, first of all, we saw microvesicular steatosis. And you may remember early on I mentioned that this was a feature of acute and chronic hepatitis C. We don't really understand the basis for this. It's quite likely this comes from mitochondrial injury that may be promoted by the core protein. But there are some outstanding questions about the pathogenesis of this, which, as you can see here on this slide in the lower right, is evidenced really well with an oil red O stain. And these are all fat droplets within the hepatocytes. We also found hepatocellular cancers in these mice. Not only this lineage, but multiple lineages. And this is a 13-month-old male, and these were all in male mice, just like humans have much greater risk of liver cancer if they're male rather than female. This is a quite large nodule, actually, that is in fact hepatocellular carcinoma. And these are some pictures of different tumors that were taken from different animals in this lineage. And we saw this not only in this one lineage, but in several lineages. Now, there are probably 3 or 4 papers in the literature now that discuss and demonstrate that expression of HCV proteins in transgenic mice can lead to liver cancer. What distinguishes this lineage in this report is that it was expression of the entire viral polyprotein and at a low physiological level. There are reports of core transgenic mice also developing hepatocellular cancer, but in that case, the level of core was grossly overexpressed and easily detectable by Western blot, which was not the case in these mice. So we believe these mice are probably more physiologically relevant. Now, interestingly, if you looked at these mice at an earlier age and you stain them for BRDU after BRDU exposure, in other words, identifying which of the hepatocytes are actively engaged in DNA synthesis, you can see that the transgenic mice had a number of, had an increased proportion of cells engaged in DNA synthesis, presumably proliferating, than the non-transgenic littermates from the same litter, although there's obviously some variation in these studies. So we have a situation then where you're expressing hepatitis C proteins, and you can see here there's absolutely no inflammation. in this case, and you're developing cancer. So inflammation is not the cause of cancer in these mice. But something related to the expression of the proteins is driving proliferation of the cell. What could that be? Well, we know now that the virus and its protein complements interact with literally dozens of different host proteins. They do so in ways, for those that are listed here at the bottom of the slide, that are important to the viral life cycle, either entry, for example, or replication in terms of these proteins over here to the right. They also do it, presumably, in ways in which the viral environment is modified, or the cellular environment is modified in ways that promotes replication of the virus. Some of these interactions, for example, are with innate immune signaling molecules, and you'll hear more about that later in the course. But a number of these interactions are with host cell proteins that are recognized to have tumor suppressor properties. Now, why would the virus want to do that? Well, the virus is evolving to ensure its own survival, and to do that, it must replicate. And the hepatocyte is a very quiescent cell, half-life probably on the order of some months, 3 to 4 months or longer. It's not unlikely that the virus has developed ways to promote hepatocellular proliferation to enhance the pools of nucleotide triphosphates that are available, for example, for its own replication. And we know from studies in cell culture that the replication of hepatitis C is enhanced by stimuli that enhance the proliferation of hepatoma cells, the cells we use to culture the virus in cell culture, and that as those cells become stationary, the level of replication of the virus drops. So I won't have time to really talk more about that aspect of it, but I want to focus on one interaction in particular that we have spent some time studying, and that is the interaction of the polymerase NS5B with the retinoblastoma tumor suppressor protein known as RB. Now, you may remember I told you a few minutes ago that E2F was an important cofactor that was reciprocally modulated and actually interacted closely with c-Myc in ways that are increasingly looking like maybe fundamental in the malignant transition that occurs between high-grade dysplastic nodules and full-blown early HCC. Well, RB is a tumor suppressor that regulates the activity of E2F. That's a transcription factor. And the story I want to outline to you is one in which we have demonstrated an interaction between the polymerase of hepatitis C, NS5B. This is the enzyme that catalyzes the synthesis of RNA. That once that complex, that interaction has occurred, leads to the recruitment of an E3 ubiquitin ligase, known as E6-associated protein, one that actually has a very important interaction that's relevant to papillomavirus carcinogenesis, and that the recruitment of E6AP to this complex leads to the ubiquitination of RB, and then the degradation of RB by the proteasome. When that happens, one has activation of E2F, which is normally repressed by RB, and cells are driven into proliferation. They go from resting into the G1S phase transition. Now, the loss of RB by the cell and activation of E2F does more than simply drive proliferation, because the RB is involved in a number of other interactions that are important for apoptosis control, replication and repair of the DNA, and checkpoint controls such as the mitotic spindle checkpoint, a point at which the cell senses whether it has sustained DNA damage and it's safe to go on to divide and create a couple of daughter cells. So we'll come back to that. So I want to show you a few slides then that just demonstrate why we came to this conclusion. Well, I think you probably heard about replicons from Steve when he talked recently. These are RNAs that can replicate within cells after their transfection and do so autonomously without integrating into the nucleus of the cell or the chromosomal DNA complement, certainly. And we studied cells that had a full-length replicon that expressed a complete polyprotein from a genotype 1b virus, such as one would find in Japan. And we found in these cells that the abundance of RB was significantly decreased, but if we treated the cells with interferon and cured them of the replicon, it was restored, as you can see. We did pulse chase experiments that showed that the half-life of RB in the replicon cell was significantly shorter than in the cured cells. And we showed also in the replicon cells that the amount of PRB or RB RNA being made was significantly upregulated in response to the downregulation of the protein RB, as it should be, as we know from regulation of RB abundance in the cell. So replicon cells then have reduced RB abundance, this tumor suppressor protein that has a pretty important influence on the cell cycle. We also showed that if you infected cells with the JFH1 virus, this unique genotype 2A virus isolated in Japan, that one found similar decreases in RB abundance over time that correlated with the onset of hepatitis C protein expression. So not only did this occur in replicons, it occurred also in infected cells. And we showed that this was associated with an interaction with the polymerase NS5B that could be demonstrated in an immunoprecipitation experiment, where when we immunoprecipitated with antibodies to RB and blotted with antibodies to NS5B, we could see a clear NS5B-RB complex. This simply controls down here. This would work reciprocally the other way also. When we looked at the JFH1 infected cells, we saw something quite unusual. Normally, RB is nuclear. And you can see it here in these uninfected cells. There's 2 infected cells here. But you can see these uninfected cells have exclusively nuclear RB, whereas the 2 cells that are infected and are staining with NS5A here have this very strange cytoplasmic extension of RB. And this is basically the RB complexed with NS5B, which is in the cytoplasm. And you don't see it in every one of these cells, but you certainly see it here. Now, this is a confocal, a laser scanning confocal microscopy image. And in this kind of image, it's actually quite difficult to assess the abundance of RB in those cells. But if you were to do a good quantitation, you would find that probably there's less nuclear RB there than there is in the adjacent cells if you set the sensitivity correctly and so forth. The important thing in this slide is that it's actually in the cytoplasm. Does it happen in people? Well, we're doing studies now to try to demonstrate that, but I wanted to show you a few early results. This is a section of normal liver stained for RB. Now, RB in this slide is red, and HCV proteins are green. But you can see that compared to HCV-infected liver, there generally is a relatively more robust RB expression than seen with the infected tissue. If you look over here to the right, you'll see examples of 2 cells that we're able to identify with conventional laser scanning confocal microscopy the presence of HCV infection. This is HCV antigen labeled with polyclonal human antibody. And you'll see over here on the right in particular this cell in which the HCV antigen overlaps very precisely with the RB antigen. And there's very little, if any, RB in the nucleus of that particular cell. To the left is another example. So this is very preliminary, but if you look hard, we seem to see in the liver exactly what we see in infected cells in culture. Now, we've shown that if you treat these infected cells in culture with an inhibitor of the proteasome, we have complexes of ubiquitin with RB that can be relatively easily identified. And we can actually find evidence of the complex between this E6-associated protein, NS5B, and RB in these cells. So I don't have time to show you all of these data. We have several papers that have described this. But what we do know is that the interaction of NS5B with the polymerase involves residues near the active site of the polymerase, which was a bit of a surprise when we first understood that. But I think we have that now quite well documented. In this region that has homology to the leucine X, cysteine X glutamic acid motifs of RB binding domains in adenoviruses and papillomavirus and other small DNA virus oncoproteins. Now, this particular sequence varies a little bit among hepatitis C strains. As you look at the whole panoply here of different genotypes, it could have either an asparagine or a cysteine in that central position, but the motif that's capable of binding RB is present in all wild-type viruses. And this simply shows you a GST pull-down experiment in which the wild-type sequence is either the N or C sequences in the middle position there are capable of a pull-down, whereas when we mutate that sequence, there is no pull-down. And again, I'm going through those data quickly because they've been published, and that particular aspect of it is not too germane to this story. Now, more recently, we've mutated that motif in the NS5B sequence and have looked at its effect on the ability of the virus to replicate in cell culture. Now, one of the hallmarks of these viruses, any small RNA viruses, is that they have evolved usually to the optimal sequence. They have tremendous evolutionary potential because in the average patient with hepatitis C infection, probably 10 to the 12th virions are produced daily. Probably every single mutant is being produced 1 million times a day in every single individual. So if there's a particular mutant that gives you an advantage, it will be selected during the course of that evolution. When we modified this motif, we saw a variable drop in the replication competence of the virus, and this was as determined by virus yield following RNA transfection of Hughes 7.5 cells, I think. You'll notice here that there was absolutely no virus produced, and that's because the mutation actually involved the aspartic acid in the GLY-ASP motif that's involved in coordinating magnesium ions in the RNA synthesis process. But you'll notice in particular the C316A single mutant and the leucine 314A mutant had significant decreases in their replication capacity, but they did grow. Now, we looked at those polymerases because one explanation for the decreased replication of those variants, those mutants, was that the polymerase obviously is mutated. Maybe the enzyme itself is not functioning well. When we did that and we focused on the C316A mutant, there was absolutely no difference in the ability of that polymerase to elongate or de novo initiate RNA synthesis. Furthermore, when we isolated preformed replicase complexes from the infected cells, so these are heavy membrane complexes that contain the actual replicase complex that's involved in making the viral RNA, and then quantified for appropriate and similar amounts of the NS5B polymerase or the NS3 protease component of that complex, we found that the mutant actually was about 3-fold more active in making RNA ex vivo. So that leads you then to the suggestion that motif is probably controlling some aspect of the intracellular environment that is important to the permissiveness of the cell for replication. And that gets back to the RB regulation. When you look at the C316A mutant, you can see that it totally fails to regulate RB compared to the wild type that downregulates RB. So the logical explanation then would be that the virus has lost the ability to regulate RB, and this is detrimental to viral replication, and that's why we have lower yield. Now, interestingly, in the course of those experiments, we noticed that the leucine 314 alanine mutant regularly reverted to a leucine 314 valine. I think it was originally, well, it was a leucine originally at that position. So you can see that the alanine to valine mutation leads to about a 30-fold or more increase in virus yield. And when we looked at that valine mutant, we saw it had regained the ability to regulate RB. So here you can see DAPI staining of infected cells. This is the nuclei, obviously, of these infected cells. And here you can see with cells that are infected with the wild-type virus, you simply can't see the RB in the nucleus. Same over here with the leucine 314 valine mutant. Whereas the leucine 314 alanine mutant that doesn't regulate RB, there is robust continued expression of RB in all of the nuclei. So it would have been nice if we could have gone on and knocked down RB with an siRNA approach and shown increased replication of the virus, but those experiments simply didn't work. We saw decreased replication of the virus, which was a bit of a mystery. But we suspect that an siRNA approach, where you knock down the cell cycle regulator with RNA silencing, is like a sledgehammer compared to the fine-tuning of RB that the virus is likely to do through its expression of NS5B. So that leads us then to the question, does NS5B regulation of RB actually attenuate E2F transcription factors? Does it actually alter the activity of E2F? Because that would be important for carcinogenesis, since E2F overexpression in transgenic mice leads to cancer, and it acts cooperatively with c-Myc, as we've seen. Well, the answer is yes. If you simply overexpress NS5B in cells, you see an increase in promoter activities of promoters that are controlled by E2F, in this case, the promoter for MAD2. Now, MAD2 is a protein that is critical to the mitotic spindle checkpoint. And in the presence of either overexpression or underexpression of MAD2, transgenic and knockout mice are prone to aneuploidy and the development of hepatoma and liver cancers. And you can see quite clearly there's an upregulation of the MAD2 promoter with the two wild-type forms of NS5B, but not with NS5B where the RB domain has been knocked out. You can see also that RB message is increased in the replicon cells compared to the cured cells. So all that fits. Now, interestingly enough, very recent work in the lab that I'm not going to show you has shown us that MAD2 protein expression is actually significantly suppressed in infected cells. So in addition to this transcriptional upregulation, there is in fact translational downregulation or altered stability of MAD2, which could also be important to carcinogenesis. Well, I'm going to wrap up here with just a few more thoughts. If you look at mice in which you acutely or chronically ablate the expression of RB, you find dysregulation, either upregulation or downregulation, of several hundred genes. It's not a very simple surgical change that you've made to the cell, but you have profoundly altered the expression in the cell of a wide range of proteins, some of which are, in fact, involved in control of cell proliferation, the classic RB function, but others that are involved in protein catabolism, for example, or innate immune responses. These are also represented among this panoply of RB-regulated genes. And among those genes are genes that are important for DNA repair and genes that are important in the checkpoints, the multiple checkpoints that determine whether a cell that has injured DNA complement goes on to divide and have daughter cells or not. For example, the mitotic spindle checkpoint. So we put together a sort of overarching hypothesis, if you would, in which we suggest that infection leads to the expression of multiple viral proteins, of course. And this leads to the downregulation of RB. Now, I've shown you data for that, but there are also data that suggests that these proteins interact with other tumor suppressors, such as DDX3, for which there is a role been suggested in breast cancer, or DDX5, a dead box helicase that's a critical transcriptional coactivator of P53, or P53 itself. And that these interactions that downregulate tumor suppressor or proliferation control proteins are designed to enhance the cellular proliferation in ways that will favor replication of the virus. And of course, that's a positive feedback loop that leads to greater expression of the viral proteins. Now that in turn will promote the kind of host responses that we have seen with inflammation and mitochondrial injury leading to the microvesicular steatosis. And that will lead to oxidative stress, which over time, with associated inflammation, will certainly be a pro-carcinogenic factor. However, the ablated activity of these tumor suppressor proteins also impairs the activity of DNA damage and mitotic spindle control checkpoints, so that the cell that is now exposed to an environment that's rich in oxidative stress and in which it is prone to accumulate DNA damage is now unable to recognize and handle that damage, and inappropriately goes on to divide in the face of chromosomal injury. And that leads over time then to progressive DNA damage and instability of the genome that result cumulatively in the evolution of liver cancer, as we discussed at the outset. Now, another view of this, of course, is this interaction of E2F, which is regulated by RB, and c-MYC, which has been shown recently to be profoundly important in this late-stage malignant transformation of liver cancer. And one could postulate that the NS5B regulation of RB leads to increased E2F activity. That actually downregulates the apoptosis that occurs as a result of c-MYC overexpression. And that simply favors this late-stage malignant transformation of the high-grade dysplastic nodule to early hepatocellular carcinoma. At least that's a hypothesis that we think is worthy of further investigation. So I'm going to stop at that point. I'd like to acknowledge two people in the lab, three people actually, that have contributed to some of the data from my lab that I've shown. One, David McGivern, who's done the recent RB mutants in the background of our cell culture competent HCV. Yuchong Liang, who's done the microscopy I've shown you today. And also Subhasa Munakata, who first identified the RB interaction, is now at the University of Tokyo in Akio Nomoto's department. So with that, I'm going to close, and I'd be happy to take any questions if there's any time left. Thanks very much for your attention.